ближе к проекту.
Будет ли ведение какого-то?
Да, видеозапись.
Представление.
Презентация?
Сейчас.
Представите.
Окей. Уважаемые друзья, коллеги, представляю вам Чарльза McKenna, профессора университета Южной Калифорнии, который великолепно говорит на русском языке. Вот. И вы будете лекцию делать на каком языке?
На английском.
На английском. Хорошо, но на вопросы вы сможете, если что, ответить на русском.
Для вашей практики.
Я надеюсь, что у нас с лабораториями, где работает Чарльз McKenna, и наши в будущем будут налаживаться взаимодействие по научной и образовательной части. И эта встреча у нас не первая лекция, не первая. Завтра Чарльз McKenna также выступает на конференции FISTECH-Bio в пленарной сессии и в сессии разработки инновационных лекарственных препаратов. Поэтому всем, кому тематика интересна, также добро пожаловать и завтра. Ну а сегодня я желаю вам удачного выступления, удачной лекции и вам интересных знаний. Задавайте вопросы, ответы.
Thank you very much. And so I will present this lecture in English. I'll try to slow down a little bit, but I also want to say that if you prefer, если вы предпочитаете задавать мне вопросы ваши на русском языке, тогда пожалуйста. So my lecture is entitled Chemistry at the Service of Biology and Medicine, and it has two parts, and between the two parts is a break. So our topic today, the first part has to do with bisphosphonate nucleotides as enzyme mechanism probe. So this is chemistry at the service of biology. And one of the take-home messages from this part is that chirality counts. Chirality is very important in ligand recognition of nucleotides in a way that could have implications for drug design. And secondly, in part two, we will talk about prodrugs, in which we take pretty good drugs and we make them even better drugs by chemical modification. So this is chemistry at the service of medicine. The picture is supposed to show the discovery of the element phosphorus. And it's kind of interesting, this is, if you will, in the 17th century, the professor, and he's made this great discovery, and he's thinking the Nobel Prize won't be for another 300 years, so too bad. But if you can see in the back, there are a couple of graduate students who are not paying much attention to their professor. So I'm going to start off with a few, shall we call them, not quite so serious slides, and then we will get into our chemistry. The idea of medicinal chemistry really began at the start of the 19th century with the work of Paul Ehrlich in Germany, who created the idea of the magic bullet. So this would be a compound that magically would strike whatever was causing disease. It could be a cancer cell, it could be a bacterium which was pathogenic, it could be a virus, although he didn't know what viruses were at that time. And it would be highly selective. It would not harm the organism that was hosting the target of the magic bullet. So this was the beginning of chemotherapeutics. And it was also Ehrlich's contribution to do the first real screening. Now, yesterday I visited Chemrar, which has a chemical library of over 1 million compounds. But it was Ehrlich who had the idea first to take a large number of compounds, in his case, a bit under 1,000 compounds, and test every compound. And then try to see some kind of pattern. And in his case, he was looking for a compound against the disease syphilis, which was a particularly important and dangerous disease in Europe around 1900. And the compound that he found that was most effective was the 606 compound that he tested. And so this compound was called Salvarsan 606, the first effective anti-syphilis drug. And on this German banknote, Deutsche Mark, which preceded, of course, the euro. Not only is there a picture of Ehrlich, but if you can see on the left, there's a picture of his drug. And it's actually a correct structure. So the key here is the magic bullet, our drug, it must be toxic to the target. It must be more toxic to the target than to the host. And we'll measure this selectivity by the therapeutic ratio. The EC50, the effective concentration, needs to be as low as possible, and the toxic dose, the LD50, needs to be as high as possible. So the implication for this at the level of chemistry is a difference in binding ability to the target enzyme of the pathogen relative to a similar enzyme in the host. Now, before we get into the real lecture, I just want to say a word about the fact that here's a chemist on money. I feel personally, above the Nobel Prize, the highest honor is to have you and your work put on a banknote. Everybody in the country and in international business looking at you, looking at your molecule, wondering who this is. So here's some other examples of this phenomenon. These are French banknotes of the pre-Euro era, and particularly pay attention to the Curies. So you see that both of them got on the note. And if you look carefully at this note, there's various images related to early X-ray mobile wagons. You see up in the upper left, alpha particles and so forth. And on the reverse side, you can see there's some kind of traditional classical distillation setup. In this other banknote, Louis Pasteur, on the reverse side, there's a whole series of interesting things. There is what's supposed to be a chiral crystal. You could argue whether it's really a correct representation of a chiral crystal. Here's vaccination. Here's the microscope to observe the enantiomeric crystals of racemic acid and so forth. So, since we're here in Russia, we're wondering, what about Russian chemists? Have they ever had such an honor? I don't know about the money, probably not, but at least on stamps, which is not a bad honor, we had this representation of the chemist Prodrug, who did very important chemistry structural work in the 19th century. In Kazan, he also has a beautiful statue, but I think that the stamp is a better honor. Just one last word about Prodrug. He also has a Facebook page. So I think this is a rather interesting token to his representation. The reason I mention him particularly is in American chemistry textbooks for organic chemistry students, Prodrug has 2 or 3 pages in chapter 2 or 3, Introduction to Structural Theory of Organic Compounds, typically. So American students have heard of Alexander Butler. A last remark about fame and chemists. We can go to SciFinder and just look up at citations of an individual or a particular paper, as you know. Now, here's a citation pattern from Linus Pauling. Linus Pauling, in December 1952, published a paper in the Journal of the American Chemical Society for the structure of DNA. At that time, they already had a Nobel Prize for chemistry. He would get a Nobel Prize for peace. So this, if he got this right, would be number 3. The only person to ever win 3 Nobel Prizes. And this is the graphical abstract. And you can see all the bases are on the outside of the DNA and all the phosphate groups are in the inside. So this was what we would call an unfortunate screw-up. And look at the citation pattern. 1953, there were a few, and then people stopped paying attention. Around the time he died, there was one big spike, and then there was a little bit later on. Let's compare this to the correct paper of about 4 months later by Watson and Crick, and you can see this is a fairly well-cited paper, but considering this is one of the most important papers in the history of biology, it's not that many citations. So what I'm leading up to is your very own Professor Falken has a much better record than Watson and Crick, in a way. So here is Valeri's record for his click chemistry paper 2002. And on the committee, you can see that he's doing a lot better than Watson and Crick were doing. So hopefully that will lead to yet more glory for Valeri. So I just make these preliminary remarks also because this is a long way of introducing the fact that Valeri Falken has just joined our university, as was announced yesterday officially, and he will be my colleague starting July 1st of this summer. So now let's go to our actual topic. DNA replication is mediated by a whole series of polymerases, replicative DNA polymerases, copy DNA, but DNA has to be also archived, it has to be preserved, and it's subject to damage from light, damage from exposure to reactive chemicals, and for other reasons. And so there exist DNA polymerases whose role is to repair DNA. They find a mistake and they fix it. Polymerase beta is a small enzyme which has this function. It has a molecular weight of 39 kilodaltons. And it's important in a pathway called the base excision repair pathway. This is an enzyme which is not very capable with respect to fidelity because it has to operate on a base which is already the wrong base and replace it. It is a polymerase that is a favorite for us to study because there is a whole series of crystal structures of this particular polymerase, and because of its small size, it's easy to manipulate and to study. An important motivation from a translational point of view to study this enzyme is that it is now recognized as a potential new cancer therapy. Now, it might seem paradoxical. Why would you want to inhibit the enzyme that is repairing DNA, and yet cancer is fundamentally a disease of damaged DNA? And the reason is that the cancer cell typically overexpresses this enzyme because if it becomes too damaged, it has to do a bit of repair to continue to be a viable cancer cell. And in particular, drugs that act against the cancer cell by damaging the DNA, such as cisplatin, can be countered by the cancer cell by overexpressing this enzyme and repairing the damage done by the drug. This leads to drug resistance. So the thought is that a drug that inhibited this enzyme, specifically targeted to the cancer cell, could help to overcome resistance of DNA-damaging anti-cancer drugs. How does this enzyme work exactly? So if you look at the upper right here, I don't know if we have a pointer handy. We don't. All right. So up here at X, we have a damaged base. This DNA has been damaged by some means. So in the base excision repair pathway, the first thing we do is we cleave a phosphate bond with sugar. So we cleave this bond here to, in this case, it's to an A. This is not done by the polymerase, but by an enzyme glycosylase. And then Paul beta inserts the correct base. So the other strand is a C, it will insert a G. And then it has a second activity, lyase, which can be used to make this bond. We go down here, and in the last step, we excise the bad base. So, Professor Myron Goodman, a biologist, and myself have received a grant recently from NIH to study the mechanism, structure, function, and fidelity of this enzyme using a whole array of probes, including X-ray crystallography, theory, biochemistry, genomics, and other methods. So our basic idea of how the enzyme probably works in its active site is as follows. So here's an incoming, in this case it's a DUM MPP, so it is a non-hydrolyzable analog. Here's the triphosphate part of the molecule. Alpha is closest to sugar, beta, and at the end is gamma. There are two magnesiums involved in the active site complex, and there are several active site residues that bind the magnesium and also hold in place the terminal primer 3' hydroxy group. It is this group which will attack at the alpha phosphorus and displace pyrophosphate to make the insertion of the incoming correct nucleotide. So this being the case, it means that if this step, what we call the chemical step, is the rate determining step for the whole reaction, then we know from organic chemistry there should be a leaving group effect. In the rate determining step, that is the transition state, this group is leaving, or breaking this phosphorus-oxygen bond, then the nature, the stereoelectronic properties of the pyrophosphate will affect the rate of the reaction. If, on the other hand, there is some conformational change, which is the actual rate determining step for the mechanism, it won't matter what this leaving group is. So it's a very simple idea. So to probe this mechanism, we have synthesized a very large array of analogs in which we replace either the alpha-beta oxygen between the alpha and the beta phosphorus by a carbon, which can be substituted by methyl, halogen, so forth, to modify its properties. Or alternatively, we can replace the beta-gamma, gamma-beta, this is the beta-gamma oxygen, by a carbon. Again, it can be substituted to modify the leaving group properties. And this creates what we call a toolkit. That is to say, an array of these compounds that have a broad diversity of stereoelectronic properties that can be used to explore the effective structure and mechanism. These are some of the compounds that comprise the toolkit. So we have di and monofluorinated, simple methylene, dichloro, monochloro, fluorochloro, and so forth. And of course, these are made not just in the DG series, but in all four bases for the four nucleotides as well. Now, to look at the effect of the leaving group, we need some kind of quantitative parameters. So what we use is the pKa of the bisphosphonate that's leaving. So the pyrophosphate normally leaves from the natural substrate. In these analogs, a bisphosphonate is leaving. And that bisphosphonate will have 4 ionizable protons. We're interested in the 4th one, so we would take off this one, this one, this one, and measure this one, called pK4, therefore it's the 4th proton. We determine that pKa will be determined by substitution on the carbon. So obviously, electron withdrawing halogens make the pK lower, methyl groups make it higher, and so forth. And so then if we plot the enzyme turnover rate as the log of the rate constant, it's plotted here at the y-axis, versus the pK4 of the leaving group, what we would expect to find would be, if there's no leaving group effect, the rate doesn't change. But if there is a leaving group effect, there will be a plot such as this one. We can see that as we make the compound more acidic, meaning the corresponding conjugate base is more stable, then we have a better leaving group, the rate is going up. And so these plots tell us immediately that indeed the chemical step is the rate limiting step. PO bond breaking is the transition state event in turnover by this enzyme. There are 2 other things that we can point out here. One is that there is some separation between these symmetrically substituted dihalo compounds and monohalo compounds or unsubstituted compounds. And secondly, if we have the incorrect base, that is to say, we're putting in the wrong base on purpose. You can see, first of all, the rate goes way down, which is what you expect. The enzyme rejects the wrong base. But also there's a much bigger separation between these dihalo nucleotide analogs and the monohalo nucleotide analogs. So I'm not going to get into the current thinking about why we get this difference, but I want to emphasize instead a different point. If I can make it happen. There we go. If we put a monosubstituent into these beta-gamma methylene bisphosphonate analogs, in this case we put in one fluorine, we create a new chiral center. And since the nucleotide is already in an enantiomeric compound, this means we actually generate diastereomers. In these diastereomers, the carbon-fluorine bond will have one of just two orientations, which will be very well defined inside the active site. So if we carry out modeling, which is being shown in this movie, and green here is the fluorine atom, this is the nucleotide modeled into the active site of DNA polymerase based on the enzyme active site structure. What we find is that for one of the two isomers, there is an interaction possible between an arginine, a guanidinium group, which is down here in blue, which is not possible for the other diastereomer. And that's calculated to be worth maybe a kcal, 1.5 kcal. It should be worth about a factor of 5 or 10 in the rate. So it means the enzyme should be able to distinguish between these two isomers based on a completely new interaction, namely that between this fluorine atom and the guanidine of the active site arginine, which is called R183. So what you're watching in the movie, while I describe this, is the actual docking data that led to this conclusion. So the prediction then was that this new interaction could cause selectivity by the enzyme for one but not the other of these isomers. When you synthesize these compounds, essentially all you do is take a bisphosphonate and you attach it to the mono denucleoside monophosphate. And so this means you always get mixtures of the isomers. Fluorine is in both orientations. So to test this hypothesis, first of all, the X-ray crystal structure was determined where the protein was exposed to both of these isomers to see if it had any preference. The prediction was it would have a preference. And as it turned out, in the crystal structure, you only find one isomer. So the protein does distinguish between these two. Why is that important? This is important for two reasons. First of all, there's a lot of controversy about what is exactly the bond or interaction between a carbon-fluorine fluorine atom and active site moiety, such as arginine. Fluorine is said to be a group that typically interacts with arginines present in many drugs. And the nature of this interaction is really not very well known. Here is a case where we can establish the exact position and alignment of this group and compare it with moving the fluorine over here, where no such interaction is possible. But maybe more importantly, this particular arginine is specific to the repair enzyme DNA polymerase beta. If we want ultimately to have a drug which is based on a nucleotide structure that would be selective, we want it to be selective for this enzyme and not able to bind to other polymerases. And so this is a kind of proof of principle that you could utilize groups designed to interact with arginine 183 in polymerase beta to increase selective binding. If this is true, perhaps not only is one diastereomer selectively bound, but perhaps it's also turned over selectively as well. In other words, the transition state energies are different. So we take a mixture of the two isomers obtained synthetically, we put in a very large amount of enzyme, and just let the mixture turn over. And if there is no selectivity, we should get an equal amount of the two isomers reacting. We can follow this by NMR, and the NMR result shows that one of the isomers reacts faster than the others. So here you can see, here's the starting spectrum. Here's the spectrum after a few minutes of reaction, and clearly just one of the isomers is reacting more rapidly than the other. So the enzyme selectively turns over as well as binds one of the isomers. To look at this phenomenon further, we would need the actual individual diastereomers. And it did not prove possible to separate them by all the methods we tried. I won't go through the long list. This chiral fluoromethylene group is actually rather far from the neighboring chiral sugar carbons, and so the effect of those carbons on this prochiral center is not very big, not big enough for the methods we tried to do separation. We'll skip that slide, and that one, and that one. And so that led us to consider, can we actually synthesize a nucleotide which has a chiral carbon at that position? So to do that, we take a bisphosphonate and we prepare a double chiral diastereomer mixture, which we make a morpholity derivative on this side. We bring in a chiral benzyl group here, which has the carboxyl groups, this mandelic acid. That also is changed to a mandelic acid amide. And that compound in 3 steps can be converted into one or the other of the appropriate diastereomers. This is because this compound can be separated by ordinary HPLC into its 2 diastereomeric forms. So we published this in the JCS a couple of years ago. This was the first synthesis of a chiral methylene substituted nucleotide. I'll just briefly go through the synthesis done by PhD student Yue Wu in my group. So we start with, let us say, X equals fluorine, a simple fluoromethylene bisphosphonic acid converted to the tetramethyl compound. That compound can be selectively cleaned to remove a single methyl group with sodium iodide. We can take that unmasked phosphonic acid group and esterify it with mandelic acid in the form of its own methyl ester, to form this derivative. Now we can remove the methyl groups, but not remove the mandelic ester by treatment with bromoyl trimethylsilane, followed by methanol hydrolysis, giving this acid. And then we take off this methyl group with sodium carbonate and decorate this intermediate with a pair of morpholines. This compound can be separated by HPLC. The reason we put the morpholines on is that if you don't, you cannot separate this intermediate into its diastereomers by HPLC. Once you have those individual diastereomers, it's an easy matter to take off the morpholine from the phosphoramide side, as shown here. Then we react it with the morpholino derivative of the corresponding monophosphate of the dinucleoside that we're interested in, of the D-nucleoside that we're interested in, to form this protected intermediate. Then we use conventional hydrogenolysis to remove the protecting groups, and we have our final compound. And it turns out that these individual compounds, as we had already seen in the enzyme experiment, have discrete phosphorus and fluorine NMR. So you can actually follow their behavior individually by either phosphorus or fluorine NMR. So here, for example, is a set of spectra for the monochloro compounds. This is the mixture, so you can see the two signals. The individual stereoisomers have one signal. Now, a curious thing is that when we expose crystals of the DNA primary and template complex with Paul beta to each of these isomers, in each case, it formed the ternary complex. Now, previously, we saw that the enzyme selected one of the two isomers to form the ternary complex crystal. But if it doesn't have the choice of the isomer it wants, it will accept the other isomer. So it means that selectivity is not an absolute selectivity. And we confirmed that the individual isomers have different turnover rates with polymerase beta for nucleotide base incorporation, and that, again, these processes can be discriminated and followed by fluorine or by phosphorus NMR. With the individual nucleotides available, we can measure the binding constants to the enzyme. They are different depending on the stereochemistry. They are much larger, in other words, weaker binding if you have the wrong base, but they are still different depending on the stereochemistry. And all of the rates of turnover are diastereomer structure dependent. We've also adapted this approach to many other chiral holomethylene nucleotide analogs, such as the alpha-beta CH ATP, which is being used to study kinases. This is a rather different method used to do this synthesis, in which we start with this protected nucleoside as the tosylate. Notice the 2', 3' hydroxy is also protected and reacted with the appropriate chloromethylene, in this case bisphosphonate, to form this diphosphate analog. We then introduce a amino acid derivative as the chiral auxiliary. These then do separation of the nucleotide into its two isomers, and then proceed with conventional methods to take us to the desired nucleotide. So we have made alpha, beta, and beta gamma chloromethylene ATP diastereomers, and potentially these are probes for a huge array of enzymes that utilize ATP. So I may have gone a little more quickly than I intended, but let me just say a few words about our laboratory at the University of Southern California. It would be nice if we were located over here or maybe over here, but this is actually Malibu. This is Santa Monica. University is close to central Los Angeles, which is about 15 miles to the east of this spot here. The university was founded in 1880, so it is the oldest university in California. It is symbolized by the figure of the Trojan. So this statue is at the center of the campus. And one of my duties at the university, in addition to doing research, is to be the Dean of Sciences. And so we took this picture in front of the administration building, just as a symbol of that. Another comment I want to make about Los Angeles is that I think many people consider it to be a city in which the car is king and people don't walk, there's no public transport. But in fact, it has a metro, which is expanding. It even has a cable car, which is called the Angel's Flight. costs 25 cents. And this, to give you an idea of where the university is located, this picture was taken from the top of one of the university buildings looking to the north towards the mountains. So I've gone a little quickly, but we can stop now and see if you have any questions.
Well, can I ask you, you synthesize alpha, beta, and beta gamma phosphonates.
Right.
What is better for biological reasons?
Well, in a polymerase, you have a nucleotide. You're going to cleave the PO bond between the alpha phosphorus and the beta phosphorus. Your leading group is pyrophosphate. So this means if you put the carbon between alpha and beta, it's not a substrate, it's an inhibitor. If you put it between beta and gamma phosphorus, it's a substrate. For a kinase, you're cleaving the POP bond between the beta phosphorus and the gamma phosphorus. So in this case, it's the reverse. The beta-gamma methylene compound is the inhibitor, the alpha-beta compound is the substrate.
And one more question, where I saw very complicated fluorine spectrum, NMR spectrum. A lot of splitting. Why?
Well, I don't know. There's a lot
here.
of splitting.
No,
This one?
Yes, this one.
So, first of all, this is a fluorine spectrum of the mixture of the two isomers.
Oh, it's mixture. Okay.
Does that help? So for each of these compounds, assuming that the diastereomers have discrete spectrum, you have a doublet from this phosphorus, a doublet from that phosphorus. It's proton decoupled.
And proton coupled or not?
These are proton decoupled.
Decoupled.
Decoupled, yeah. And so this is why we're assigning the outer piece to the different diastereomers. But that's confirmed by mixing the individual diastereomers. It's rather interesting, by the way, that just a couple of years ago, a chemist named Jakeman in Canada published a paper in which he said he made a similar type of compound, and the fluorine spectra for the isomers is completely indistinguishable. And he had spectra to prove this. So it means either we made a big mistake or he made a big mistake. There's only two possibilities here. So we investigated his compound. We published in Journal of Organic Chemistry last year that if we make his compound, the spectra are completely resolvable.
But phosphorus spectra are distinguished for both.
That for fluorine. Phosphorus, only certain resonance, not every single resonance. But the problem was that he did not control his pH, and so you get these sharp lines only above about pH 9.510. So if any of the oxygens are protonated because of exchange, it tends to broaden the lines out. So he was right, but he was not right. Any other question? So is everyone clear why these compounds are proving that the rate determining step is a chemical step? So if this is correct, if the hypothesis is correct, if we could find a different polymerase for which other people have adduced evidence that shows that the rate determining step for that polymerase is conformational, not chemical, with our toolkit we should get a flat line. So that will be a very important support for this method. Now, there is such an enzyme that for which everyone working in the area has predicted it must be a conformational mechanism. And so we have already started experiments on that enzyme. But maybe next time we can present those results. So how would you use this kind of tool to study the enzyme further? If you change the conditions of the enzyme, could it be that you could change the rate determining step, for example? And so that's another thing which we are looking into. And then finally, is it possible to take advantage of the interaction with the arginine to actually create a selective drug for a translational purpose? And that's yet another project that we are currently working on. So, any other questions? So we can take a break if you like for 5 minutes, and then we'll resume with a different topic. This structure, so you can see it's cytosine, and then the normal sugar has been completely changed into a 3-carbon group. The central oxygen, originally hydroxy on that, at that position, which is a chiral center, is esterifying hydroxy, is making an ether linkage rather to hydroxymethylphosphonic acid. And so the phosphonic acid is standing in for phosphate in this case. This is a compound which is widely used in the clinic. It's an intravenous drug only. The compound was discovered by Antonin Poli in Prague, the Czech Republic, at the Institute for Organic Chemistry and Biochemistry. And this is still one of the most potent drugs against double-stranded DNA viruses. These viruses include orthopox viruses such as smallpox, cowpox, vaccinia, variola smallpox. And for this reason, this is a compound that has been considered as a potential biodefense compound in case there was a terrorist attack involving smallpox. The reason that this compound actually would be completely useless for that purpose is that it has very low oral bioavailability, less than 5%. And this is because at physiological pH, the phosphonic acid ionizes and therefore acquires a negative charge, and so it won't pass from the intestine into the bloodstream. And so forth, therefore it can only be used intravenously. So if this was a drug you wanted to administer to the entire population of nearly 300 million people in 24 hours, it would not be practical. Therefore, it's of interest to try to find a way to make this compound an oral drug. And for that, we need a prodrug version. So I don't know if you're already highly familiar with the concept of a prodrug. Does everyone totally understand what a prodrug is already? No? Some do, some don't. The idea of a prodrug is you take an existing drug, you modify it to change its pharmacology. For example, now you can take it orally. And that would enable it to also perhaps reach its target in the body better. But there has to be some chemical or biochemical process that removes the pro group and reactivates the drug once it's reached its target. Now, one could argue that perhaps the first prodrug was aspirin. In the history of aspirin, aspirin started out as salicylic acid, which started out as a kind of herbal remedy, the willow leaf, which proved to be useful for headaches if you boiled it and drank the infusion. In the 19th century, salicylic acid was isolated and found to have efficacy against headaches and pain in general. But this is a compound that has a phenolic group in it, and so it's very irritating to the stomach lining. In And so the bio company came up with simple chemistry to acetylate this phenol group and created aspirin. Aspirin is much less irritating to the stomach, and once it reaches the blood plasma, the acetyl group comes off and you're back to salicylic acid. So this is why if you take a bottle of old aspirin tablets and smell it, it has a vinegar smell because of some decomposition of the prodrug. There's something not too widely known, I think, is the bio company in the 19th century, at the very end of the 19th century, did something we see very commonly today in drug companies. So the drug company will find some structure that works and is good on the market. So we'll try to copy it, repeat it, or make a variation of it. And so the chemist who worked on aspirin was aware of the fact that at that time, morphine was known for its enormously beneficial pain-killing effects, but also feared because of its enormously addictive qualities. So the thought was, why don't we acetylate the morphine and we'll make a better compound, maybe it won't be addictive. And so, and we already know how to acetylate, we did it for aspirin. So they acetylated this phenol group and this cyclohexyl hydroxy group and came up with a new compound called heroin. And for a while, this was a compound, you can see it's used in cough medicine, cough drops, cough lozenges of every kind, sold in the drugstore over the counter, like aspirin. You could just go and buy it for your cough. Good for bronchitis, whooping cough, and so forth. Heroin is a cough medicine. And it was only after a few years people realized heroin is even more addictive than morphine. It's even worse in that respect. And it became an illegal drug. So the problem we have with the antiviral compounds at Othavir is we needed to be able to penetrate membranes, to get from the intestine into blood plasma, and to get from the plasma through the cell membrane into the infective cell to reach the virus target. Now, membranes generally are permeable to small hydrophobic molecules like O2, carbon dioxide, N2, and so forth. Small uncharged polar molecules like water, ethanol obviously, is a very effective compound taken orally. Certain larger uncharged polar molecules like amino acids, glucose, and nucleotides are not able to penetrate membranes. Ions, which have a positive or negative charge, are not able to penetrate membranes. So in the case of our antiviral drug, we have an ion, which is also a larger uncharged polar molecule, not able to penetrate lipid bilayers or membranes. So our first idea was maybe we could take advantage of the fact that there are many transport mechanisms designed to transport amino acids or glucose or other sugars through membranes. And so we decided that we would esterify the phosphonic acid of the drug to the hydroxy group of an amino acid in perhaps a di or tripeptide, and thereby hope to create a prodrug. Now, the advantage of using an amino acid for this is obviously amino acids are not toxic. They have functionality that you can tune. This is a natural linkage, the PO amino acid linkage. Serine, threonine, or tyrosine is a natural linkage, and so we can expect there will be enzymes that are endogenous in the cell that will cleave it off and release the drug. And in general, this type of ester bond is stable under gastrointestinal conditions in the stomach, pH 2.3, and in the upper intestine, pH around 6. So in our early versions of this, we simply took a peptide. In this case, it was a serine-valine peptide with appropriate protecting groups on it, synthesized the ester using that DIEA and PBOP, and deprotected to create our prodrug. So in our prodrug structure then we have serine. We left the methyl group on here to make it uncharged. Here's our PO linkage. This will have a positive charge on the valine. Valine has a hydrophobic side chain, so this could also help penetration of membranes. But the real idea here was that it's already known that there are transporters in the gut that will transport small peptides, and so we were hoping that by hooking the drug onto the dipeptide, it would simply be transported through mimicking a natural dipeptide. The peptide transporter that we were targeting is called PEPT1, and this is a transporter that transports peptides coupled with proton transfer. And there is a model compound used to study it called Glycar, which, as you can see, is a dipeptide analog, if you will, of glycine and methylglycine. And so when we compared our dipeptide with the prodrugs, we made one from alanine and serine, one from valine and serine. And the ability of these compounds to inhibit transport of the model dipeptide, Glycar, is in the low millimolar range, but it is certainly better than the parent drugs themselves, which are in the somewhat higher millimolar range. And using a classification system of affinity to the transporter called Brandeis classification system, these values put it in the medium, put our prodrugs in the medium affinity area. So it seemed that possibly these compounds could work. When we study the interaction of these compounds with the transporter, we use a model system, namely the oocyte or the egg of a frog, which is connected to a two electrode voltage clamp. And if there is transport, this system generates a current which is concentration dependent, depending on the concentration of what is being transported. And this is not a very readable slide on this projector, unfortunately, but essentially, if you have a compound which is really transported like Glycar, you get a result like this, and the parent drugs are not transported at all, which is not surprising. There are structural requirements for this transporter for its substrates. And we have to distinguish between compounds that have an affinity for the transporter and compounds that are actually transported across membranes by the transporter. First of all, the compounds that are transported should be di or tripeptides. Our compound are dipeptides, so that should work. And they work much better than amino acids or hydropeptides. Secondly, the transporter likes a free terminal and carboxyl and amino function on the peptide. There has to be a peptide bond, obviously. It prefers L-amino acids in the peptide over D-amino acids. And it always prefers at least one amino acid in the dipeptide, which has a hydrophobic side chain, so that would be something like alanine, valine, isoleucine, leucine. HPEPT1 seemed to be an ideal target because it functions with relatively low affinity. It has a very high capacity, so it's a very efficient nutrient transporter. It could translate a lot of drug from the intestine into the plasma. using this proton gradient. As we said, it mediates trans-epithelial transport of dye and tripeptides, and it's widely expressed in the intestinal tract, so it's in the pancreas, bile ducts, even in the kidneys. So at first our results looked encouraging. Here is an experiment using the frog oocyte. So this is measuring how much current we get at a given concentration. First of all, shown on the green bar here, this one going here, for Lysar. And then we add one of the antiviral drugs unmodified. Then we add Prodrug constructed from D amino acids, or a mixture of D and L amino acids, or from both L-valine and L-serine, or from L-valine and D-serine. And what the results show is that these two dipeptides are very effective in shutting down the transporter, so the conclusion was at least they are binding quite well. So these seem to be the ideal dipeptides to use in our prodrug. Here again results the control, where we simply put in Lysar at high concentration, at lower concentration, and the various drugs and prodrugs. And again, we seem to be able to completely shut down transport with a number of these different dipeptide prodrugs. The only problem turned out to be that if we look for the actual current generated by the prodrugs, it turned out to be zero. So in other words, they're binding to a transporter, they inhibit normal substrate transport, but they themselves don't get transported. We also found that in the blood plasma, the half-life of these dipeptide prodrugs was rather short, maybe an hour or 2 hours. And so after a couple of years of work on this, we were forced to abandon our whole concept of a dipeptide transport-based prodrug approach. Now, if in the United States you were funded by the National Institutes of Health, they're paying you money, you promised you would get a result. So if you don't get the result, you face a decision. And so we decided that the only way to save this project was to throw away the old concept, explain to NIH that it didn't work, and try something different. So what we decided to do was to abandon the whole idea of trying to achieve a prodrug targeting transport and just make a prodrug able to be transported. And our approach then was to go from a dipeptide to just a simple amino acid and to focus on modifying that single amino acid to modify its pharmacological properties, in particular to make it more hydrophobic and more stable. And what we ended up with is this new generation prodrug, which we call AMP tyrosine-based prodrug, in which we have a single tyrosine as the prodrug component, in which the nitrogen is a carboxamide, and on that nitrogen we'll put an alpha group, which can be varied in length, obviously, to tune the hydrophobicity of the compound. So in this way, we can get the optimal balance of lithophilicity, solubility, bioavailability, efficacy, and potency. To synthesize these compounds is quite simple, and we scale up the yield of these compounds, and make them in grams. Now the question is, do these compounds actually work? So first of all, we did a transport study for the L-tyrosine ester of the cyclic form of the adenine version of cytophager called cyclic HPMPA, which has a carboxamide modified with an isobutyl. This is a comparison of the concentration of the prodrug you can achieve by just infusing it directly and by supplying it orally to fasted mice in the mice model. And you can see that the concentration peak that you can achieve orally and by infusion is very similar with this prodrug. But with the prodrug, the persistence in the plasma is quite a bit longer than with the original drug, which means that its effectiveness can continue for a single dose for a considerably longer time. From this curve, we were able to calculate the bioavailability, in other words, the percent of drug that actually ends up in the plasma in the oral dose is close to 40%. As we saw before, that of the parent drug is less than 5%. So what that means is that with this compound, we've achieved the goal of an orally bioavailable form of cytophager or its derivatives. Now, as we saw before, what is important in determining whether or not such a compound is really effective is to compare its toxicity to its effect on its target. And so here is some antiviral data obtained in vitro against human cytomegalovirus, which is a herpes virus. This is a virus that causes blindness in AIDS patients, and it is also a serious systemic virus infection for immunosuppressed patients, such as patients who are under cancer treatment. So to understand these data, first of all, we look at data for 3 parent drugs, encyclovir, HPMPA, and the cyclic form of HPMPA. These are the adenine version of cytophager. The effective concentration against the virus, which is the concentration that reduces the number of viral plaques by 50%, are in the micromolar range in vitro, which of course is not particularly good. The cell toxicity of these compounds is in the upper micromolar range, and so the selectivity range is between 9, which is not very good, and 240, which is actually pretty good. Now, here's some data for the prodrugs. And let's go right down to this one, where we have the prodrug consisting of the tyrosine ester, carboxamide, with a C16 alkyl chain attached to the carboxamide nitrogen. What we can see is that the EC50 has now gone down to 8 nanomolar. The toxicity to cells has also increased, but the selectivity index is still fairly good. It's over 100. But effectively, we've taken a drug from the micromolar regime and taken it down to the low nanomolar regime. And therefore, this is a second benefit that we got with the prodrug strategy. Not only does it make the drug orally bioavailable, it also has increased the potency by a couple of hours of 10. Now, in this case, in the effort to get rid of this POH group that is left unmodified when we make the tyrosine ester, we formed a cycle, internal cycle of the drug. It turns out for synthetic reasons, because this is going to create a chiral phosphorus, it would be preferable to have the acyclic version of the drug. So we also made the acyclic prodrug as well. We go down to the red entry in the table. Again, this is a C16 modified carboxamide, tyrosine derivatizing the acyclic form of the drug. And now the EC50 is even better. It's close to 1 or 2 nanomolar. And again, that can be compared with the acyclic form of the drug, which is 400 nanomolar. So it's increased the potency by a factor of over 100 again. And here the selectivity of the prodrug is considerably better than that of the original compound. So this Prodrug approach seems to be very promising, and we are currently extending it to virtually all of the different DNA viruses, and we are in the process of applying it to several other viruses, such as, in particular, HPV as well. So our conclusions are that, first of all, the tyrosinemate Prodrug platform is significantly enhanced A and B drug potencies, in fact, for all the DNA viruses we've tested. And secondly, I haven't shown data for this, but they are able to inhibit viruses with a serotype-dependent SAR. What that means is that for many viruses, if you take a group of infected patients, they have genetically different forms of the same virus. Those viruses have different sensitivities to a particular antiviral drug. And finally, these prodrugs provide a dramatically enhanced oral bioavailability. So we now have got NIH grant support to try to develop these compounds in preparation for taking them into a phase 1 clinical trial. And I think what it shows in this project is that if you have a scientific approach and a hypothesis which is unsuccessful, you should not give up, but you have to be brave enough to rethink what you are doing, and in particular, try to keep your grant support going for that particular project by some new ideas. And so, in this case, that has turned out very well for us. So, just to conclude, I want to mention some of the people who work with us in our own group. One of the interesting things about our group recently is we have more and more Russian people as group members. So I have Dr. Boris A. Kashimirov, Boris Anapolovich, a postdoc, Maria Zakharova, and there is a new student who's not listed here named Ivan Krivov. Our work on the nucleotide analogs was done in collaboration with Professor Goodman at USC, his students, crystallographer Dr. Sam Wilson at NIH, and his collaborators. Persons doing antiviral testing for us are at the University of Michigan Medical School in a company called TSRL, Dr. John Brock, Dr. John Goldinger, and other research we've done on bisphosphonates supported by Procter & Gamble Company or the Chilcott Company in collaboration with the University of Oxford and also with Harvard's Massachusetts IND here. So I think with that, I will stop and ask if you have any questions.
Questions? First, in your book, any pharmacodynamic measurements other than concentration for your project? For example, water distribution or clearance?
Yes, you're talking about PK measurements? Yes. Those were conducted not in our laboratory, but in the laboratory of our collaborators at the University of Michigan.
Second question. I assume that this program serves for mobilizing the urine or the bile?
You mean how's it excreted?
Yes, yeah.
In the urine.
Can I ask you? And my question about water solubility of these, especially cyclophosphamate with long acyl chain.
You know, that's an excellent question, I have to say. So this is, if you will, a weak point of these compounds. Obviously, with the C16 hydrocarbon tail, they are not too soluble. And so they're formulated in different ways. One way it's in DSO, and then it's diluted in solution. we're also looking at just an aqueous dispersion of it as well. Because it is potent at very high potency, which means you don't need so much drug, that actually countervails against the solubility problem to a considerable extent. Nevertheless, that does pose some problems for dosing.
And one more question, just my curiosity about aspirin. You said that it's very nice to block groups, but not carboxy group. My question about aspirin. Did somebody try to block carboxyl group in salicylic acid?
If you're asking the historical question, I don't actually know the answer to that, but I'm sure they did because they were making lots of compounds. But I think that was a very good example. Very often in synthetic chemistry, you will make compounds because you can make them. And so you already know how to take an acid and alcohol and make an ester, so you make it, right? But it also shows that using this method of making drugs, where you simply make a compound and try it, you can have many unfortunate surprises. And in fact, the chemist who worked on aspirin was the same chemist who proposed to apply this chemistry to morphine to make heroin. Notice heroin comes from the word hero. So this was supposed to be a heroic compound that would vanquish pain. And he himself, to set a good example, was taking this compound to show it was safe, and he became seriously addicted to heroin.
And about heroin, just now we cannot buy arsenic antidote. It's forbidden in Russia.
Can you buy heroin in the drugstore?
No. Well, it's so-called the precursor of some bad compounds.
But that is yet another interesting phenomenon of how a drug can be, just as a drug may be a prescribed drug but gradually becomes accepted as a non-prescription drug, so a drug like this ends up as a completely illegal drug once it was sold like toothpaste or aspirin. That's an interesting phenomenon. Phosphorus itself as an element was once considered a very important drug. And if you look in Pharmacopoeia, Chepnik, go back a few hundred years, you can see at first phosphorus, just the element white phosphorus, is being recommended for all kinds of diseases, which is very useful for. But over the years, the dose is always going down, which is obviously driven by toxicity, right? And just before it finally was taken out of the Pharmacopoeia, the dose is extremely low.
This was your compound with the problem of species different pH values for different species. Usually take for mice, right?
Correct.
And what about the pharmacists, for example, non-human primates? Is it the same? Very often in disorder concept, when it's converted to the active compound, very often the effect which you observe with small animals, small vertebrates, is not the same.
This is true. And this is the reason why the original dipeptide compound was considered ultimately not so promising, because it had a half-life in the rodent model of about an hour or two. These compounds, depending on the length of the alkyl chain and so forth, can have half-lives of 10, 12, 15 hours in the model. So that gives you some safety when you finally put it in humans. None of these compounds have been in humans yet, obviously. Other questions?
Do you think, do you have any suggestions about if we want to approach certain tissue specificity, for example,
Well, this is another quite interesting question. So, I mentioned at the beginning that these A and B antiviral drugs, such as cidofovir, HPMPA, and so forth, have kidney toxicities. I didn't show data, but the prodrugs are showing a different PK distribution, and in particular, they have not such a high concentration in the kidney. Now, perhaps your real question is, can you target the compounds better? For the viral infections that we're interested in, the virus tends to be fairly pervasive, and so we don't actually want to target just the liver. If, for example, we were targeting hepatitis virus, then it would be advantageous to remodify the probiotic to get targeting into the liver, which probably will be possible. Any other questions? No? Well, thank you very much.